Hello, I'm Danil Zilov and I'm first year master student of SCAMT Institute of ITMO University in St. Petersburg. And I will talk about Pannopi, Prokaryotic genome assembly and annotation pipeline. With the development of next generation sequencing technology, the volume of genetic data is growing as rapidly as its cost decreasing. Now it's necessary to identify and automate areas of analysis that are routine, time-consuming, and likely to contain random errors. For this purpose, we created Pannopi, flexible, scalable, easy to install and use pipeline for large-scale analysis of bacterial genomes. Only sequence reads are needed to run the pipeline and to get a report. This approach is useful as for biologists without experience in bioinformatics to obtain analysis quickly and without intermediaries, and also for bioinformatics because our pipeline contains a basic set of proven and effective tools with the possibility of flexible customization. Pipeline is a program consists of a sequence of commands and tools. It helps to automate the work, save time, and eliminate random errors. Pannopi is made up with the help of SnakeMake workflow management system because it's, first of all, connected with the Conda package management system, which contains almost all tools that needed to run pipeline. And also SnakeMake based on the Python programming language, and this allows users to Python computing alongside with the running of tools. Usually, the process of analyzing bacterial genome sequencing follows these steps: data preparation, genome assembly, structural annotation, and functional annotation. Pannopi follows all of these steps with the quality control and some additions. All starts with the data preparation. Reads go through trimming and optical duplicates leading to preparation quality control. Quality control organized with the FastQC and Jellyfish tools. FastQC used to get standard statistics of reads quality, Jellyfish for k-mers and coverage counting. Jellyfish histogram visualized with the help of GenomeScope tool. Next, prepared reads move to the assembly and filtration stage. On this stage, genome is handled with the help of one or few assemblers. Users can choose which assembler to use. If there are few assemblers, then scaffolds go to quality control stage. Sample with a higher number of N50 moves to the filtration stage. Filtration goes with the help of our own Conterra tool. It parses blast analysis of assembly, looks for contamination, and removes it if any. In addition, it determines the closest reference taxon ID and downloads reference assembly and annotation. Structural annotation stage based on the two annotators, Proka and PGAP. Users can choose which annotators to use. If there are both annotators, then QC started. Sample with the closest to the reference number of bases, CDS, rRNA, and tRNA genes move to the pangenome building and functional annotation. Pangenome builds with the help of Rory tool. It compares annotation with the all prokaryotic annotation of proceed bacteria taxon. For operons identification, we use our approach based on colocalization from the string database, a distance between genes, and no similarity of biological functional through the gene ontology database. Best annotation files from previous stage moves to the ignocmapper tool for functional annotation with gene ontology database, PySpy tool to find Profac genes, Abricate tool to find antibiotic resistance and virulence genes, and MLST tool to determine MLST type. Then all output files are pulled together in a report file. As the report of the pipeline work user gets genome assembly file, QC files of sequence reads and assembly, annotation in a range of formats, functional annotation report, and the closest reference genome. 24 samples testing dataset were created to check out pipeline running. Full analysis on our system takes from 91 to 186 minutes depending on pipeline configuration. Now the Pannopi is on a testing and integration stage. We are building web interface and finishing pipeline assembly. Expected release date is this summer. So thank you for your attention. If you have any question, I would be happy to answer by email. You can find it below or use the QR code to contact me.